Thanks for the introduction. Today I'll be discussing the next step in the evolution of library preparation. Before I introduce the next product, I wanted to take a step back and discuss some of the underlying conceptual principles of library preparation, which we use to guide development. The first step in improving library quality is to ask the right question. People who make libraries every day are generally focused on input and output. And so for them, the primary question is whether or not they can make a library with their available material that is appropriate for their sequencing application. What we came to understand is that it's not about whether or not you can make a library, but how do you know that you made the best possible library from the available material? In other words, is your library the most diverse, and does it yield the highest and most uniform coverage? To answer this question and really drive effective product development efforts, a good proxy for library quality is needed. We know from our work with library amplification that final library years aren't really good predictors of sequence quality, because for one reason or another, there's a disconnect between the input into library preparation and final library yields. If you dissect the library construction workflow into functional units, it is clear that the core library construction process, so the process by which randomly fragmented DNA becomes adapter ligated, is really what determines the number of unique library molecules, whereas library amplification only generates duplicates. So to evaluate the intrinsic efficiency of library construction, it's not the final library yield, but really that intermediate step, that post-ligation library yield, that becomes a good indicator for what's really happening. This, of course, assumes that your library amplification is well optimized and performed with a low bias enzyme. And because absolute yields of post-ligation libraries aren't very useful when comparing across different inputs and sample types, we define this term conversion rate. This is essentially the percentage of input material that becomes adapter ligated library. Using this normalized metric, we were able to benchmark performance, and that really guided our efforts towards making those performance and workflow improvements over the past few years. So when we start to plot the evolution of library construction, which is expressed as the maximum conversion rate for 1 microgram of high quality input, the picture looks something like this. The baseline prior to the introduction of the Kapa library preparation products was less than 1% of usable material. We saw a 10-fold improvement with the generation of the Kapa library preparation kit, which was accomplished with relatively minor improvements to workflow and formulations. With the Kapa with bead library preparation kit, we saw an additional 4-fold improvement. This allowed the core construction library process to be performed in one tube. And then with the development of the Kapa HyperPrep, we saw conversion rates reaching 60%. This had huge benefits for low input samples. Having addressed many of the bottlenecks associated with library preparation, including the ability to accurately QC and quantify input material, the conversion of input molecules to appropriately ligated molecules, and the efficient and low bias amplification, along with the ability to accurately quantify library yield, the only remaining process to tackle was fragmentation. In line with our approach to date, we realized that our fragmentation solution had to be high throughput, it had to be enzymatic. Like the HyperPrep kit, it had to be versatile and able to support library preparation with a variety of inputs, sample types, and for various sequencing applications. It had to support high library construction efficiency, and it had to introduce very little bias. We are very excited to announce that such an enzymatic fragmentation reagent is now available. Shown here are bioanalyzer profiles with 100 nanogram of input material, ranging from human genomic DNA to PCR products. They were sheared for various times with our enzymatic fragmentation reagent. Phenotypically, they're very similar to that of Kovara's shears. The mode is very consistent across all times and samples, irrespective of the input and complexity of your input DNA. The most exciting part about this particular workflow is not that we've developed a truly usable enzymatic fragmentation reagent, but that it's fully integrated into the HyperPrep workflow. This allows us to perform fragmentation in one tube, fragmentation all the way down to post-ligation cleanup. It allows us to further streamline library preparation so that library prep is accomplished in 1.5 to 3 hours. And it's also flexible, just like the HyperPrep kit. The elimination of loss during mechanical fragmentation and the transfer into library construction, along with the synergistic effect that we see with the HyperPrep kit and the enzymatic fragmentation reagent, allows us to have even higher library conversion efficiencies. For inputs of 100 nanograms above, we're seeing efficiencies approaching 100%. This opens huge doors for PCR-free workflows, targeted sequencing, and any other application where you might need high yields of libraries. And there aren't just benefits for higher input ranges. We also see these benefits at the lower end of the spectrum. For 1 nanogram of DNA, we can see conversion rate up to 20% of that into usable library molecules. This means that a number of samples that would have failed library preparation and with other chemistries can now be processed in a way that doesn't allow you to amplify large amounts. So high library construction efficiency is an obvious benefit, but with enzymatic fragmentation reagents, a key question is, what about bias? Start site bias is one way to quantify fragmentation bias. And we're showing that data here for a human exome sequencing experiment. The libraries were made with 3 different workflows: Covaris plus HyperPrep, HyperPlus, and a tagmentation-based approach. Looking at the first few bases near the start site, we see that the HyperPlus nucleotide content is a little less uneven than that of the gold standard Covaris plus HyperPrep, but it is much more even than that of the tagmentation-based approach. The good news is that the minimal start site bias doesn't impact the overall coverage uniformity. Shown here are the coverage uniformity plots for the same human exome capture experiment, and what you want to see is a narrow distribution around your coverage depth, which is 20x in this case. There are a few obvious observations. The first is that there's no significant difference between Covaris and HyperPrep and HyperPlus. Whereas on the other hand, the tagmentation-based libraries, you see 2 distinct differences. There are coverage hotspots as well as low and lumpy coverage. The result is that you get a lower overall mean coverage, meaning that you need to sequence more to get your desired coverage for every base. Another example of the benefits of HyperPlus is evident when you look at GC bias plots. This data was generated during microbial whole genome sequencing of varying GC content organisms. And as you all know, you want to see a normalized coverage of 1 across all GC bins. In this study, 4 workflows were assessed: HyperPlus, Covaris plus HyperPrep, tagmentation, and a competitor that also utilizes an enzymatic fragmentation methodology. You can see that we have relatively even coverage with HyperPlus and Covaris, whereas there is a large underrepresentation of AT-rich regions with the competitor and tagmentation-based approach. We see a similar pattern when we look at GC-rich genomes. We see more even coverage with the Covaris and HyperPlus workflows, whereas we see a large overrepresentation of the GC-neutral sequences with the competitor kit. For an AT-rich genome, we see near-perfect results for Covaris and HyperPlus with a normalized coverage of about 1 across all GC bins. In contrast, both the competitor and tagmentation samples have uneven coverage. It appears to favor the more GC-neutral regions at the expense of the very AT-rich ones. So to briefly conclude, with the HyperPlus kit, we have streamlined library preparation in a single tube from fragmentation through ligation. We optimized a low-bias, robust enzymatic fragmentation solution. We created a workflow with the highest available library prep efficiency, and we utilize our Kappa Hi-Fi enzyme for high efficiency and low bias amplification. The result is an industry-leading solution with the speed and convenience of tagmentation-based product. but significant benefits in terms of robustness, flexibility, sequencing quality, and cost. So with that, I'd be happy to answer any questions. We're at booth 36. Thanks.